I've had a bunch of these inventions, and they were fun, but they weren't really big deals. And when I was at Southwestern, I started to realize that that I really wanted to do science differently and organize it differently, how we did these things, and I wanted to go after some really big deals. And so I started organizing my center around just the basic idea that Neil was outlining. Rather than a traditional academic, I wanted to pick out a big problem, see if I could come up with an invention to solve it, then have the ability to put teams together to see if that solution actually could be true. Now, this is just what business does, actually. They got a problem, they figure out how they're going to solve it, and they put a group together and they figure out what the product's going to be. The only thing I wanted to do differently was have this be a disruptive technology that we'd put together, not just putting two pieces of metal together and make a product. I wanted to really transform stuff. So I came over here, talked to Mike Crow, talked to George Post, who they recruited here, which I thought was an amazing recruit, if they could get somebody like him to head up this institute. And I thought, rather than just be tolerated over at Southwestern, I want to come here and be appreciated. And so because this is exactly what they were saying they wanted to form this institute to do. And there was no other institute like this, doing this kind of stuff in the country. And then they also bought into this idea that I wanted to take about 3 years, to have enough funding in 3 years to go after some of these big concepts that I had, that I thought were really off the wall, but if there was any validity to them, they could transform things. And one of them was this idea, I've already gotten some hassle about this, about cancer eradication. That isn't a promise, that's a goal. It's just like when we said, Apollo, we're going to put somebody on the moon. You had to say you were going to put it on the moon. If you didn't get to the moon, you didn't do it. But at least that's what your goal was. And that's what our goal was here, is that I had an idea how possibly we could treat cancer like infectious disease and eradicate it, just like we've done with some infectious diseases in the past. And what we have are basically 4 types of Apollo projects that represent those large areas that Neil told you about before. But this is the one I'm going to tell you about, how far we've gotten with this concept of this Apollo project. But everything that we do, everything I do, is based in physical economy. I first figure out what is the problem, and then try and figure out how do we solve it. And the problem that I think everybody recognizes is healthcare. And cancer is certainly part of that problem. And I'm just going to quickly go through the physical economy part of this, because some of you may be impressed by the numbers. One is that our total healthcare expenditures are about $2.2 trillion a year in the United States. That's projected to be $4 trillion a year by 2016. As a percent of our GNP, it's gone from almost nothing to about 18.9% last year. As a percent of our total GNP, it's projected to be somewhere between 25, or maybe even 30% by 2015, depending on what happens here. That's a huge amount of our GNP being presented just for healthcare. In terms of total spending, healthcare far outstrips energy. Even in the last blurb of energy expenditures, healthcare was still more expensive than energy, and it's even more so now. So it's our biggest spending effort. And that wouldn't be so bad if it was a very productive investment. But it isn't, because we spend most of our healthcare in the last years of our lives where we're not productive parts of the society in terms of contributing to jobs and things. So we're spending a lot of money, but we're not getting a lot out of it. And the projection is that that's only going to get worse, because by 2050, we're projected to have about 20% of our population be 65 years or older. Other nations have a totally even more skewed demography than that. So we got a problem here. What is the problem? Well, we spend about $2 trillion a year on healthcare, drugs, diagnostics, care. It's interesting, when I ask people what percent of that is spent on drugs, I get numbers like, how much? 15? 50? 30? That's about the normal average. I think the average is about 50 usually in most crowds. And it is too high, because it really is about 10%. We spend 2% on diagnostics. We spend almost everything on patient care. That's what we're spending our money on. So if we want to do something about healthcare, we have to spend it. We have to do something about this part of the cost. So what are our options? Well, one is we can do what we offer. We just don't give as much healthcare anymore. That's what we can do. But the other thing is we reduce the costs. And our vision is that we do this by transitioning from a post-symptomatic medicine to pre-symptomatic medicine, that we take care of things before they get bad. And maybe more than that, we make things even better than good, if possible. So this is the idea that we have to try and do. There's a very interesting human evolution aspect of this that is amazing. Human beings are doing something that no other species has done before. Every other species, including ourselves up to a few hundred years ago, had what's called a survival or quality of life curve that looks something like this. You're born, some of you die, you go along pretty evenly, and then you die quickly off. So if you're a lion, you break your foot, you're dead. Very simple, right? And that's largely what the life curve that people had up until a couple hundred years ago. But as we got better about taking care of ourselves, our life curve now looks more like this, where there's a long decline. That's a very expensive long decline. Somehow, I'm not sure how, but somehow we need to square that curve again. We need to have it so people have a longer, productive life and then die in their sleep. And it's all over quickly. And in fact, if we really did this right, we'd move this out here a little bit, have a longer productive life and die in our sleep out there at 150 or something. But how do we do that? And I think one of the ways that we begin to square that curve is by making medicine more, as Neil was calling it, proactive, that we prevent things before they happen, and we now start to take a lot of this chronic disease part of this out of this picture. Now, where does cancer play in this? Cancer is about $250 billion a year direct costs in the United States out of that $2.4 trillion. So it's about 10% of the total cost. Now, these figures used to blow people away. Up until about a couple months ago, $250 million. You were just, amazed that we were sending. That's nothing anymore, right? That's just, I don't know if some of you may have entered that old politician, Durskin. Give me $1 billion here and $1 billion there, and all of a sudden you've got a lot of money. Well, that has gone by the wayside now. It's now give me $100 billion here and $100 billion there. It's still a lot of money, even by today's standards. So it's 10% of our total healthcare costs. And that's what we're targeting right here in terms of the technology we wanted to develop. What would be the advantage of making a significant effect on cancer in the United States? Quite a bit for every one of us, even younger people, but this is a benefit curve that has been developed by the University of Chicago to show the benefit to each individual for a 10% reduction in death rate from cancers. More importantly, now here we might even get to some impressive figures. If the cost benefits of curing cancer at 1% level is about $500 billion, it's about $5 trillion at 10%, and it's $50 trillion over the next 50 years if we had a cure for cancer. So even by today's standards, $50 trillion is a lot of money. So this is something we really, and that's net benefit for the society. So, but there's another aspect of this that's often underregarded, and that is that most of the cancer deaths in the world do not occur in the developed world. They do not occur in the Western world. Out of the approximately 7 million deaths a year of cancer, 5 million of those are in the developing and underdeveloped countries. And so this is a worldwide effect. And if you could look at something if you look at the measures of the impact of cancer on countries in terms of their economic impact, it's almost the same in these countries as it is in ours. So this is a serious worldwide problem. This isn't just a developed world problem. But we don't do anything about it, pretty much. So for instance, the Gates Foundation has spent $0 on cancer in the third world, even though cancer kills as many people in the third world as the three leading infectious disease causes of death, and where they've spent about almost $1 billion on that. But we spent 0 here. Why have we spent zero there? Well, the reason why is that cancer treatment costs too much. Typical year's treatment for Herceptin or Avastin are far outside the bounds of ever using that in a third world country. So if we're going to actually do anything about cancer, it also has to be something that's cheaper. We can't continue on the pathway we have now where we develop things that are productive or effective even, like Herceptin has been, but cost too much. And this is going to become an increasing mantra in the whole United States, if not the world, is that you can't just look at effectiveness, you have to look at cost effectiveness for any biomedical development that you're looking at. Okay, so everybody recognizes cancer as a problem. These are slides from the NCI itself. The data is from the American Cancer Society, but these are from Anna Barkers, who's the deputy director. So the problem that people recognize with cancer is that it kills a lot of people. A lot of people get it. It's a cruddy disease to get. It's affected everybody in this room probably. And in that recognition, then President Nixon started the war on cancer in 1973, I believe it was, and since then we've spent over $86 billion through the NCI itself on trying to mount this war. And there's a lot of criticisms, rightly so, for how this war has been mounted, especially since there's been almost no net benefit in terms of the death rates of cancer over that 30-year period. So anybody that looks at those numbers will say, where's the beef? Right? I mean, if you have that sort of investment portfolio in a business, you say something's wrong with our investment portfolio, right? So, and the NCI recognizes this, and they're doing all sorts of things to try and find a way around this conundrum. And they're getting a lot of help in terms of what they think should be done in terms of getting around this conundrum. And I'm sure you've read articles like this, many of you, about what's wrong with our war on cancer. What's the prospects? Well, they don't look so good. Frankly, if you go and look at all the trials that are in the pipeline now, there's about 1,800 drug trials and about 100 vaccine trials. The promises of effect of these are almost exclusively minor. They're measured in months, not years, in terms of the projected effects. So the pipeline does not look too good. So what does look good? What does look good is that there's a transition in concept of how we look at cancer. And this is to a large extent driven by the genomics and proteomics revolution that's been taking place. The idea is instead of treating cancer as we typically have done as a post-symptomatic event where the tumor is recognized and then we use chemotherapy or radiation or surgery to remove it, we're trying to move to a stage where the treatment is pre-symptomatic, that we develop biomarkers or other sorts of ways to tell where the tumor is very early on, and now we're still going to use the same old drugs, but they're going to be a lot more effective because we're going to use them much earlier, and the side effects will be lower. So this is a very admirable transition, and it's important to do. And in fact, we recognize this. We're trying to participate in this. And for instance, that technology that 20 of you bravely participated in out there was a technology that we're also developing. We're trying to use that for pre-diagnostic detection of cancer. It's very in its early stages, but we have developed a new type of array where you can put a drop on, literally a drop of your blood, like we took today, and then we put it on there, we wash it, and what we can measure are basically all of the antibody signatures in your blood. You have 10 to the 7th different antibodies in your blood at a particular time. They all have different features. We can measure that profile on one array. And the idea that we're trying to do is that by looking at what's called a heat map, we'll be able to tell what your diagnostic situation is, and in particular in this case, relative to cancer. Because as you'll learn about this in a minute, the cancer actually produces an immune response. Now, heat map. I'm going to teach you 3 things tonight on science that you can use at cocktail parties. One is heat map. Sounds good, right? It's a really simple concept. All it is that when we take your drop of blood and we put this on here, your antibodies will bind differentially to this spot. You have 10 to the 7th antibodies. They will bind to different spots on here differentially. So we can actually, and the antibodies are labeled so they shine, they light up. So we can look at each spot, and there's 10,000 spots on the arrays that will run for you, and each spot will produce a certain intensity of light. And we can just rank them. From 10,000 to 1 is how much light comes off of that. So if we look at subject 1 of you, and red is a lot of light and black is not much light coming off of that spot, we can just rank all 10,000 of those spots for their light intensity, right, for you. Now if we take the next person and we look at each spot, it will have a different kind of light intensity off of each spot. And so red being where they have a lot. So if you could look here, here's a person, you did not have very much antibody there, but that person had a lot. Down here, you had no antibody there, but they had quite a bit. And each one of those defines your signatures. And so when you put these all together, they form what's called a heat map, basically from hot red to cold black on this slide. And this is a very common strategy for basically signaturing your diagnostic. So we can do that, and this is a heat map comparing 600 people with different conditions across here. And if you kind of cross your eyes, you can see there's patterns in here. So these are women with breast cancer. And you can see that they kind of group together, and their patterns look different. Their heat map looks different than people that have valley fever. That look different than people with influenza, look different than children with asthma. And so we can begin to look at those patterns of 10,000 things and begin to see, okay, can we see a pattern of complexity that correlates with breast cancer early versus somebody that just has valley fever? So that's the basic concept. This is a new invention. This is relatively new. So you're the first public people that have partaken in that. So what we're trying to do then here is develop this as a pre-diagnostic system for detecting cancer. So this is a blow-up of part of that map. And in this case, this is the same thing. Here's spot number 1, 2, 3, 4, 5, 6. And these are cancer patients, and these are normal age-matched patients. And you can see that there's a difference in pattern. But it's not all black and white, right? It's a complex signature that you're going to have to look at. Same thing down here. This is another set of people. These are cancer patients. These are normal people. And you can see the differences in intensity of these spots. And that's the signature that we're looking for to try and pick that up as a pre-diagnostic. The beauty of this system, that it's so simple. I mean, 20 of you know how simple it is, right? It was literally a drop of blood is all we need to run this assay. The question is, how informative can it be? We think from our early work that it can be quite informative. And so the system we envision having is something called tandem DX-RX, DX for diagnosis and RX for treatment. And so the idea here is that people on a regular basis would take a drop of blood, put it onto one of these signature systems, and do it on a regular basis because it's so simple and inexpensive. It costs us maybe $20 to do the whole thing on one of those arrays that you did. That you could do that on a regular basis. And so we would always normalize you to yourself. You would always be looking at your signature against your own signature, which is not what we do for biomarkers. We look at you against the population. And so you do that on a regular basis, and when we see a signature that indicates that you might have cancer, we would start at least do conventional monitoring and treatment. Give you the standard of care at that point and just do careful watching. And if not, if the advanced sort of concept here is we not only see what you are reacting to, we actually make a vaccine out of what you're reacting to. We know what your tumor is doing wrong, and we personally vaccinate you. So we make that into a personal, immediate vaccine. That's the vision of this. Yes?
Do you have a different signature plate for each type of cancer, or do you assume that there's enough similarities that one plate is covered all?
By the way, I don't mind interrupting. Ask me any question, and that's why I put this whiteboard here. If I need to, I can go over there. That's a good question. So the question is, does every cancer have a different signature? We're new into this, and so far, every cancer that we've looked at, brain, breast, and pancreatic, where we have any breadth of analysis, they have their own distinctive signatures. So we might know what that signature is, say that looks like a breast cancer signature, but we know it's not a normal signature. And in some ways, the treatment doesn't matter because it's your immune system and it goes after it anyway. So that's good, and that I think we could be potentially helpful in this trend towards early diagnosis, but that's not really what I was envisioning. What I was envisioning is that we really treat cancer like an infectious disease. You don't get it at all, or at least you don't know you've had it at all. I mean, you get infected after you've had a flu vaccine. You just don't know you've had a flu vaccine, usually, if it works well. So the idea here now is, could we move this to the next phase? So we actually give people vaccines so they never know they have cancer. Is that possible? Now, vaccines have been around. People have been trying to do therapeutic vaccines, vaccines after you know you have a cancer, for over 20 years. And there's a lot of pros and cons. The pros are the vaccines have minimal side effects, unlike chemo or other therapies. They're long-lasting protection. The disadvantage is it hasn't worked. So other than that, things have gone quite well. And in fact, there was a very telling, cathartic article that came out a few years ago where all these guys would do small enough studies that they fool themselves into thinking maybe it was working, but when they put all of their work together to see if it made any difference, it made no difference. So we haven't done very well. But now we know why. And this is the second thing I'm going to teach you about, tolerance. And I don't mean in a biblical sense. This is an immunological sense. The reason why therapeutic vaccines did not work is quite simple. When you get an infection, or even when you get a vaccine, you not only, whether it's you or a mouse, you not only get the foreign stuff we're putting into you, we're trying to get the immune system to work, but you also get something that's called danger. The bacteria that we give you in your vaccine also has stuff on its surface that tells your immune system that there's a bacteria somewhere. Or even when we shoot with this gene gun, even the act of shooting actually tells your immune system that something's wrong, and it starts reacting to it, even if there's not a foreign antigen or not. So it's a two-component fire alarm here, right? You have to have foreignness and danger, and then you get a strong immune response. But look what happens when you get a tumor. When you get a tumor, tumors start developing, and they're very aberrant. They're making all sorts of weird proteins. And we've discovered this, everybody knows this. They make all sorts of weird proteins. So why doesn't the foreignness get them killed? Well, it's because there's no danger. It's just quietly being foreign. It doesn't have any bacterial cell components. It doesn't have any gold bullets. It doesn't have any of those things. So there's no danger. So you only get one part of the signal. And that is tolerance. And tolerance is there for a very good reason. That's why you survive puberty. Because when your own system sees more foreign proteins, it has to know that that's just part of you, not something it should be reacting to. So when you present something in your development that's different, you form a tolerance for it because you don't want to get an immune response to it. If you do, it's called autoimmunity, and that's not good. So we now understand this is why tumors get by. The immune system is there. It's reacting. It sees the foreignness, but it also puts a brake on it at the same time that keeps it from becoming a full-blood immune response. So there's this yin-yang thing going on as the tumor is there, and it's sitting there in people sometimes for 15 years, where it's in this holding pattern between a brake and the accelerator, and then finally something happens and it breaks out or it disappears. But this is the bane of the therapeutic vaccines. This is why they didn't work. And they'll never work as we did them. Yes?
What was different about those kinds of vaccines that they didn't work? Did they not have a carrier?
Because the immune system, when you have a tumor, it's already started this tolerance. It's already turned off your immune system to the tumor. It's too late. Unlike an infectious, and the same thing as a flu vaccine. If we gave you a flu vaccine after you had the flu, it would do you no good. Because it's already rolling. There's also a numbers problem there, but it's already rolling. So this is why therapeutics didn't work. So that's the bad news. The good news is vaccines always work preventively for cancer. If you have a tumor and then you vaccinate, the mouse dies. if you take that same mouse and you pre-vaccinate it with something that's appropriate, and then you give it the tumor always lives. So it's just the order of when you do it. You do it before, you don't get a tumor. You do it after, you get a tumor. It's very simple. And this has been supported by 20 years of research. So our logic is quite simple. I don't think biologically, I just think logically. Vaccines have been the most effective medical treatment in history. They're by far the most cost effective. They only work prophylactically. There's almost no examples of a post-therapeutic vaccine for infectious disease. So obviously, we need to make a prophylactic cancer vaccine, right? Pretty simple. But people didn't do that, and people have not worked on it for a very simple reason. It's called the personalization paradigm. There's the byword now in cancer is personalization, right? And so that's true. And in vaccines, it's also thought to be true for this kind of experiment. If you take a tumor and irradiate it, so you kill all the cells and you give it to a mouse as a vaccine, and now you come and challenge that mouse with that tumor, they'll always survive. However, if you take that mouse that's been vaccinated with the red tumor and now challenge it with a blue tumor, and it can be a tumor that arose in the same tissue, from the same reason, in the same background, everything else looks pathologically the same, that mouse will always develop a tumor and die. So this is the personalization paradigm. You can only protect against a tumor against itself. You can't protect against another tumor. Well, whenever I see this kind of stuff, and this is in a textbook, whenever I see it, I always say, let's not believe it. Let's just believe it's not true, because if it is true, I can't do what I want to do, and so let's not believe it's true. And so that's what I did. And so we did an experiment where we took a melanoma tumor, gave it to a mouse, took it out, and just sequenced the genes in the tumor just to see. And what we found was, and this is your third word, frameshift. Sounds like a sexual thing, like gender shift or something like this. It's not. It's a very simple concept, is that the genetic code is read in threes, and each of those three things corresponds to an amino acid in a protein. If you put a new base in, you change the code. And at that point, from there on out, you read junk. It's just like in Morse code. If you put a dash in at the wrong place, you won't read the same thing anymore. And so that's what happens here. That junk is a new protein for the tumor. It makes it foreign. So we sequenced all those genes, and we found two foreignness in that melanoma tumor. Two of these frame shifts that are in, and there were 11 amino acids that shouldn't have been there. If we go take a mouse and we vaccinate with those two peptides ahead of time, that mouse, most of them survive. However, if we don't vaccinate with those, most of those mice die. Now that's not too surprising, right? It says, I found it in that tumor, I vaccinate it, and I protect against that tumor. So, but here's the clinch. Then we went over to a breast tumor. Remember, it was a melanoma that we'd used before. Now we take one of those foreignness was in the breast tumor. It was also in the melanoma. If we vaccinate with this, it protects the mouse against the breast tumor. If we vaccinate with the other one that wasn't there, it's not protected. So we've in principle broken the paradigm. That we found the same foreignness in a breast and a melanoma, and it will protect against both. So that it's possible that you could make a prophylactic cancer vaccine. That's what that means. So the basic idea is tumors make weird peptides. If you pre-vaccinate with weird peptides, you protect. And if some of those weird peptides occur in different tumors recurrently, you should be able to make a prophylactic cancer vaccine. That's the basic concept. So it basically comes down to this. It's not so complicated. Can we take a bunch of tumors, extract all of their proteins out of them in one way or shape or form or another, look for all of the weird things that are in these tumors, and find cases where a weird thing occurs in this tumor and a weird thing occurs in that tumor? If we find them recurrently and we add them all up, if we had pre-vaccinated with this, we would have protected against all those tumors. Simple concept, right? So it's simple to say, simple to cartoon, Not so easy to do. So to do this, this is a proteomics, genomics, informatics, all this sort of problem. We had to get all these people together to go mine the literature, to set up new immunological screens like the ones I told you about. To do proteomics, we had to spend hundreds of thousands of dollars in mass spec systems to go look at these tumors. We had to do the genomics, where you had all these tilings and all this high-end genomic stuff. And we have teams doing all of this stuff to try and find the weirdness in tumors. That's basic what they're trying to do. Can they find enough of them to constitute that vaccine? And in fact, on the literature thing, we have to go find other people's trash, because it turns out we're the only people in the world doing this. No other lab in the world is trying to make a prophylactic cancer vaccine. So everybody else is doing these kinds of things, but because they're not looking for vaccines, they trash exactly the stuff we want. So we have to go look in their trash for our gold to get this to work. And so we set up a system that once we do find these candidates, we then look to see if they would be valid as antigens before we go into a clinical trial. So it turns out about a third of the human vaccine candidates we find are also in mouse tumors. So we can go test directly to see if mouse would be protected. The other thing we can do is in vitro, we can see if we put a real human tumor cell in there, would it be killed by a cell that was activated with that vaccine? So we can test this before we go into a human vaccine. So where are we on this amassing things? Well, we keep this running tab. This isn't fully updated, but we keep a running tab of all of the variants we found, all these weird peptides, and how much coverage they would give in the general population. So if you got vaccinated with this gene itself, it would protect cancer in 0.2% of the population, et cetera, et cetera. So we just keep running them down. These would protect at 4%. This would protect at 1%. This new thing that was found causing skin cancer, a very rare kind of skin cancer, would protect 0.1%. So if you add up everything we found so far, it would protect about 10% of the population if you had pre-vaccinated with them. That's not adequate to get into a clinical trial. We need to get about 30% to get into a clinical trial. Yeah?
Are you basing that just on the percentage of the presence of those in all the tumors that you've looked at so far?
Yeah, so it's based from screens for looking at the prevalence of these mutations in different types of tumors. Now, and times something else, and that is the probability that someone in the population would present that to their immune system. That's another factor in that. But these percentages actually could be even better, though, because a lot of times when they find something in a lung tumor, they only screen lung tumors for that variant. They have not screened to see if that variant is also in breast tumors. So we're doing that to expand hopefully find some of these that are broadly prevalent. So this is our running tab, and it's getting better. The system's getting easier and easier to run. So I think even though this has been very slow, I think the next 10% will go a lot faster. Yeah?
And what time frame is that?
This thing is about 2 years.
How many?
2 years.
2 years to develop that 10%. So
Yeah, but I mean, I have to say that most of that was developed putting that infrastructure together so that we could get the system.
do you have more critical mass now?
Yes, yes. And also a fundamental discovery we had, and other people are finding this too, is that normal cells are a lot weirder than anybody thought. So subtracting out what is a tumor weirdness from a normal weirdness has turned out to be the real catch in this problem. And it's very interesting from a basic science point of view, but it's frustrating from a goal-directed thing like this. So we certainly have challenges. The biggest thing is we have to assemble enough of those antigens to make this effective, to get enough together in a basket that we can vaccinate with and protect. And we can have a pretty big basket and still make this vaccine. So I think it's doable. There are issues about safety that we'll have to address. If you vaccinate with any vaccine, where you're vaccinating people that aren't sick, you have to be very concerned about safety. And autoimmunity is one of the things people raise as a concern. I don't think it's much of a concern, but non-immunologists raise this as a concern. And then there's the problem of getting into a phase 1. When do we do this? And then how do we actually demonstrate efficacy in a population? I mean, who would take this vaccine? Now, I will anticipate a few frequently asked questions because I've been presenting this to people. One, I get it all the time. This is such a good idea. Why isn't anybody else doing it? And that's a fair question. And it could be because it's a bad idea. It could be because I'm stupid. It could be a lot of things. But frankly, I think it's because we've put so much time and effort into this problem. And the general impression is cancer is very complicated. We have to understand a lot. And when you've invested in something you're convinced it's so complicated, it's hard for someone to accept someone coming along that's outside of the field and saying, well, maybe it's not. Maybe it's simple. And so I think there's a reluctance on people to switch from a very vested interest in the complexity to a very simple possible answer. Also because it's high risk. I mean, I don't know if I'm going to be right. I could be very wrong, and it could flop on its face, in which case that's embarrassing for some people. I've done it so often it's not anymore. But so I think it is a good idea. So the bottom line is that I put this in front of oncologists, scientists, individuals that are very respected in the field. I have not heard anyone tell me why someone shouldn't be doing this. Besides risk and all these other things, I've never heard a fundamental argument against doing it. So until somebody gives me that, then I think it's worth pursuing. The other is if tumors are producing frame shifts, why doesn't the immune system destroy the tumors? Well, that means they didn't listen to my explanation about tolerance. Because remember, if the tumor starts, it turns off your immune system, and that's why it doesn't work. You have to vaccinate with the danger before the tumor arises. So the vaccine we have may not be so effective for people that already have cancer. It would probably work better for people that don't. What about autoimmunity? I don't think it's a big problem, but we'll have to test for that. When do you envision people will be immunized? Initially, beyond reproductive age would be the first population. And then as it gets a better and better safety profile, it could potentially be immunized in earlier and earlier times. How would you do the clinical trial? This has turned out to be the big bugaboo. This is what people get choked up on. This is what I'm having to deal with the most, is how would you act, even if you had something that looked perfectly good, how would you convince people to start to participate in a clinical trial to test this out? And how long would it take, and how would you do it? And this is an interesting problem. I think I have a good solution, but not everybody does. So I hired a company. This is not in progress. They completed this study. The Center for Business Model in Healthcare in Chicago to basically start looking at these problems for me. This is rather unusual for an academic to do these kinds of things, but these are serious problems that have to be addressed before we get into clinical trials. And so these people are trying to get the best pathway for that. So in summary, I don't know whether we can pull this off. When I first started, I just thought of this as something that should be done, even if it fails. I have to say right now, I'm pretty confident we can. I don't know we can, but I'm getting more and more confident that this will work. And if it does, it's important. So we'll continue doing it until we know we fail. But if it's going to fail, I want to fail as fast as possible. So, yeah.
How do the clinical trials for this differ from other vaccines?
Well, if you think of this as an oncologist, the clinical trial would be a trial where you get people that are high risk or have already had tumors. Well, if they already had tumors, they're not good for me to do, right? And so most oncologists say, we'll do a high-risk families. Well, those take a long time and are very expensive. I say treat this like an infectious disease. When we did the rotavirus trial, we enrolled 70,000 kids, and it was over in 9 months. Because you enroll enough people, you vaccinate them, do they get infected or not? I would do the same thing with cancer. Enroll whatever it takes, enroll 2,000 60-year-olds. Their incidence of cancer is 1.5% a year. Vaccinate them and wait a year and see if they got cancer. So, that's my solution, but a lot of people don't like that because it's not traditional in this space. Yeah?
Was that the reason why you said there was 20 years of vaccines that have been produced, but they haven't used them or been effective, or they just kind of sit down and shelf?
Yeah, they were tried. People tried very hard. The question was, what happened to those 20 years of vaccines? People tried very hard at those things. Now, I should say there's a bright light on the horizon for therapeutic vaccines. Knowing that the problem was tolerance, that your immune system's already turned off, there are drugs that specifically turn off your tolerance. So the people are now doing clinical trials where they go back and administer the same old vaccines, but now turn off your tolerance. And the hope is that by turning your tolerance off, it'll allow your immune system to push the yin into the yang and go after your tumor and kill it. Now, this is something you'd only want to do in a short period of time, because if you suppress your tolerance system for a long period of time, you get autoimmunity. But for a therapeutic vaccine, that may work. And some of the early clinical data I've looked at looks encouraging in this regard. Any other questions? Yeah?
Has cancer existed throughout history, and does it exist in animals?
So the question is, has cancer always been here and does it exist in animals? Yes. In terms of recorded history, of course, yes, it's existed. I don't know of examples in archaeological history of humans, but I suspect it does, because cancer is basically a numbers problem. You have 10 to the 12th cells in you. It only takes one to go wrong to initiate this process. So you're always going to have something going wrong. It's just an odds game. In animals, yes. In fact, specific strains of dogs, for instance, get very specific kinds of cancer. And each breed, even though they're very related to each other, has a specific profile of the kinds of cancer it gets. And in fact, there are 8 dog cancer institutes in the United States set up specifically to do dog cancer trials. So, yeah.
Sorry I'm a little slower, but could you explain again how you think one vaccine, I think we're still struggling on how to detect cancer in early stages. Like we have PSA for one vaccine. Can you explain again how you think one vaccine would apply to all cancer?
Okay. So the idea is really pretty simple. Here's breast cancer, here's colon cancer, here's lung cancer, here's pancreatic cancer. So here's each one. And if you look at a number of individuals in each one, and you look at their tumors, and you find that this tumor had this one, this tumor had this one, this, etc. These weird proteins. If you had put all of those together, and every time a tumor arose, you were 100% sure that one or more of those would turn up in that tumor, then that tumor would die, because you've already been vaccinated against it. So the question is, how many of those things do you have to put together to cover 100%, let's say?
And you haven't reached that point yet?
No.
And is it possible that you won't be able to?
Yes. But I would refer back to that graph that I showed you, that a 1% reduction was worth $500 billion. A 10% reduction was worth $5 trillion. So even those effects, I think, are important. So if we discovered a number of antigens that would, if you gave them to women, it decreased their chance of breast cancer 90%, but had no other effect on any other cancer, that still would be, in my mind, a worthy product. Yeah?
So the heat mapping, that's theoretically going to be able to tell what kind of cancer you have just from a drop of blood, then why not just use that to only treat what you have? I mean, I know you're saying it would hurt to give them, you're hoping to prove it would hurt to give them all of them, but why not just give them
Exactly. So the question is, if the diagnosis was for breast cancer, and you knew that breast cancer would have this array of things, why not just give them that? And I think that's a reasonable strategy. For the therapeutic vaccination, remember, you've already had it. For prophylactic, you don't know what cancer you're going to get, right? So you can only vaccinate, and it will only protect against the people that would have had that cancer.
But I mean, if it only costs $20
Right. Yes, and I think that's possible. I think I would be happy, extremely happy, if the product was a regular diagnostic test that was as simple as a blood test. And rather than colonoscopy or anything else, just simply a blood test, and it told you that you should be at least do watchful monitoring of something, or even better yet, that it gave you an indication of what you could vaccinate against. That would be extraordinary. Yeah.
Could you also use things like family history, breast cancer, for example, to select
Yes.
the population?
For the prophylactic vaccine? Okay, so so the idea is, could you bias your chances by increasing the probability of someone having a cancer? Yes, that's a good idea. The risk in that is how many of those people actually have latent cancer already. Because if they have it already and it hasn't shown up diagnostically, their immune system has already reacted to it. So it's a trade-off there. But that I agree, that's something that should be weighed. Yes?
Why can't you showed the drop of blood and it showed up very strongly on breast cancer. Why can't that be used for screening for cancer?
We think it will. I mean that's our hope. And we've only screened about 120 patients now. 120 patients right now. And these are all the patients we've screened have been diagnosed with cancer. So now there are historical samples from these, like the Women's Health Initiative and others, where they've collected blood over time, and they actually have collections of blood before they were diagnosed, anywhere from 1 to 5 years before, mostly for breast cancer, before they were diagnosed. So we are just testing those now to see, could we have told that person was going to get cancer later? And that's the big kahuna question. Yes?
On the heat map, does that say you have cancer or only that you're prone to have cancer? And if it says you have cancer, then the whole theory blows on terms of it immunizing.
So the question is, how predictive is that heat map? You know, at what stage does it, how resolving can it be in the stage that you are at, right? Is that?
It really shows you have it. You can't use the...
Yeah, you can't use the prophylactic vaccine because it can't be prophylactic, right, by definition. But what you may be able to do is go ahead and, as someone suggested, use those same antigens, but now you would have to vaccinate with that tolerant suppressive drug at the same time. Yes, it would be a treatment, hopefully an early treatment, and a treatment where your immune system can still outnumber the tumor cells. That's the other problem with these therapeutic vaccines. In many cases, the immune system just had a numbers problem. It couldn't catch up with the tumor anymore. But at an early stage, you can outnumber it. Yeah, yeah.
And you're saying those proteins are unique to those types of cancer? They're not found in healthy cells, for example?
Yeah.
You wouldn't get a response against the healthy tissue, or they may overlap in different cancers, but...
I wish this was an easy answer. But I will make what we thought that this was going to be a piece of cake. When we found something that was in a tumor that wasn't in the normal sequence of a normal person, that we would not find it anyplace, and we were perfectly safe. When we started to look more carefully in more normals and more normal, different types of normal tissues, we almost always can find a low level of this weird protein in some of the normal cells. So that is this messiness that makes this more complicated. That's the bad news. The good news is, because we have indications now that even though they're there, the immune system hasn't seen them yet. They're there at such low levels, or they're processed out by quality control, that the immune system still is naive relative to them. And that's all we need. If the immune system is naive, we can still vaccinate with these ahead of time. I don't know if that's clear, but okay. Yeah. I wish things were always as simple as you can cartoon them. Wouldn't life be, you know? Yes.
Question for you. So, do you have an idea how much of these peptides are made early enough? Like, at what level you would be able to distinguish that you have
Yeah.
cancer, or is it already so big that
Yeah.
does this allow for early detection?
Okay, so we know best from mouse models about this, where they can set up things and control things. So the question is, how early can you detect things? If it looks like the immune system starts responding to a tumor as soon as it starts. So your immune system is already seeing that you have if projecting from mouse studies, it sees that you have a tumor there. It's making these responses. They are measurable in the blood. It's just that the regulatory part's keeping it from killing the tumor. And you set up this sort of quasi-equilibrium thing. This is why in transplant patients, there's these individual cases where people have had a melanoma 15 years ago. They die, they do a kidney transplant. And as soon as the guy gets the kidney transplant, they get melanoma, they get cancer. Because that was sitting in somebody's kidney for 15 years with their system, keeping it at bay. And as soon as you put it into an immunosuppressed person, there it goes. And there's a whole series of these kinds of cases that have turned up, which are actually lend credence to this theory about what the immune system's doing. It's holding this tumor at bay, this yin-yang-y stuff, and then something happens. You get immunosuppressed, you get depressed, whatever it is. The yang takes over the yin, and you get a tumor.
Can I ask one
Sure.
question just real quick? When you give this vaccine, do you envision that you're going to have someone mention the carrier protein or some way to elicit the danger response in addition to just the peptides?
Yes. And it turns out for all of the immunizations we do in mice, we just use this gene gun.
And
That's enough. Because as soon as we've done these studies, as soon as you shoot here, those dendritic cells, the ones that are basically your immune patroller cells, go to your draining lymph node. Just the gold hitting them is enough to send them running. And so when we immunize a mouse, we use a 1/10 of a nanogram of DNA to shoot into the mouse. It's just so tiny amount that we need to elicit the immune response to get protection. So I think the immunization will be, I think that part will probably be okay. It certainly could be optimized, but I think it'll be okay.
As we learn more about stem cells and cellular regeneration, cellular crosstalk, is that going to have any implication in relationship to how you look at your thought process in relationship to these antigens, weird proteins, and all the other things?
No. Okay. Because that is important for the prophylactic vaccine. It is. Those are important considerations when you have an existing tumor. If you actually have really prophylactically vaccinated, you will kill the thing as soon as it arises, just like an infection. If you are pre-vaccinated, you won't know you were infected, usually. I mean, stem cell stuff is all very interesting, very important for therapeutics, but not for the not for the prophylactic, as far as I understand. Yeah.
Is the FDA going to slow down the development of this getting into the public?
That will be so the idea is, will the FDA slow it down? I mean, that seems like a pretty good chance. But this will be something that, and this is why we hired that company to go look into this, is because it's an unorthodox thing. It's not really a therapeutic vaccine. It's not a therapeutic, and how will they respond to this? I think in my best mind, we will do the phase 1 trial, which is a standard safety trial. And if it turns out to be safe, I think my expectation, there will be a lot of pressure to get this into people as soon as possible, because it only does you good if you get it before you get it, right? So it will remain to be seen what happens there. I just don't know. Yes?
Yeah, on the cost management ratio for perspective, you showed it earlier, I think about 70% of the cost is kind of patient paid.
Yeah.
Do you know in cancer, what percentage do you think get cancer, say below 50 versus above 50? Do you have any idea?
So the question, the cancer incidence per age?
Yeah, do you have any idea if it's mostly for the elderly or?
Oh, absolutely. In fact, the reason why why there's an apparent increase in cancer overall. There really isn't. It's just that we're having an aging population, and cancer is a disease of aging. We don't exactly understand why it's a disease of aging. It's really amazing if you start to think. We do not understand that basic phenomena.
Prostate is definitely.
In fact, almost all cancers, except childhood leukemia, for instance, they're all an aging disease. And as I said, 60-year-olds have a 1.5% chance each year of getting cancer.
Does that minimize the cost-benefit ratio? I'm in that age group. I don't want to say don't take care of it, but does that minimize the cost-benefit ratio?
It would, depending on what the therapy was. If I proposed that this vaccine was going to cost $200,000, well, we'd have to take pause, right? But if this is a standard vaccine and it costs you $100 or less, and in fact, in our mind, we want it to be like a standard prophylactic infectious disease vaccine so that those 5 million people in the rest of the world can also have benefit of this, right? Otherwise, we haven't done the job. Okay? No one's waving at me yet. Does this mean am I supposed to stop, Sally? Yes?
How much influence does something like stress have in starting this whole process of cancer starting to come up in the body? And then there's so much you hear about on television and that about attitudes, diet, and that in helping to suppress the growth of any kinds of tumors and that. How does that fit into your research?
Not directly. I mean, so the question is, what's the role of the mental state on cancer? Not directly. And I'm certainly not an authority on that. I can say that there is increasing recognition of the direct connection between the neural system and the immunological system. They evolved from the same starting point, and they've kept a history of connection in some remarkable ways. And it's fascinating, but I am not authority on human aspect of that, so I'll stay out of that. And I'm getting the shut up signal. Okay.